Hello everyone, welcome to my fourth lecture video on the chapter Principles of Inheritance and Variation. I hope everybody are following the chapter and understanding the concepts here. I can understand this chapter has quite new terminologies for you all to understand with, but I suggest students try to understand each and every small terminologies of this chapter, because this chapter is very important for your exams, both for your board exams and for the competitive exams. Also, this chapter is something wherein we try to understand ourselves here, how exactly the characters come to us from our parent, and how exactly we give the characters for the future generations. While we transferring the characters, what are the probabilities that happen here, in what ratios we transmit a particular character and all. This chapter makes something very interesting, how exactly our system works, right? And this proper chapter, genetics, and the next upcoming chapter, that is molecular basis of inheritance, both the chapters goes hand in hand. In this chapter we understand how exactly the genes and the chromosomes work, but in the next chapter we understand what are the genes made up of, how exactly the genes express, and how could we regulate the expression of genes and all we study here. Both the chapters comes under a very broad name called applied biology. Both the chapter has a lot of scope in the future generations also. And now in the previous session what we were discussing is, we were discussing with Mendelian principles and the non-Mendelian inheritance work we were discussing with. Students will carry out the same session here, that is with what happened after the Mendelian period. We have another concept to discuss for the class today, that is chromosomal theory of inheritance. Chromosomal theory of inheritance was proposed in the year 1902, that is after the Mendelian period. We have discussed that in 1900 the Mendel's work were rediscovered by three scientists, right? That is Hugo de Veris, Carl Correns and Teschmark. These three scientists took up the Mendelian work and they republished it, and that is how the scientific community of that period got to know that Mendel has worked with this. Simultaneously, students, what you also have to understand is, when Mendel was quite busy with this pea plant, when he was trying to understand with the contrasting traits how it is working and all, along with that Charles Darwin was also working for the field of evolution. Simultaneously, the chemists, that is the chemistry people, they were working and they were trying to study with the structure of how exactly the chromosomes are there and what is this chromosome and all. So here in this particular session, what we'll be dealing with, chromosomal theory of inheritance. We'll understand how exactly the chromosomes do the work, right? So first of all, what is this term called chromosome? Chromosome. Chromo means color, zome means body, that is the colored body. What happens is, when you take out the chromosome and if you stain it with little bit of small dye also, the chromosomes receives the stain and it will appear colorful. So that is why the name chromosome is being given for this particular component. Now we all know this particular structure is a chromosome. This chromosome has two parts in it, that is, if you can divide this whole chromosome into two equal halves, we get chromatids. That is, this particular one is one chromatid and this particular arm is one more chromatid. See here, this is one chromatid and this is another chromatid here. Both the chromatids are linked at a junction called centromere, or we can call it as primary constriction. This is what you have studied in the cell, the unit of life, isn't it? So this centromere is the junction where exactly the spindle fibers will come and link during the cell division. So this centromere is a place where exactly the spindle fibers come and link and they help in the movement of chromosomes. That we have also seen in the chapter cell cycle and cell division. Now moving ahead, we all know that we have discussed already that the chromosomes are present within the nucleus of a cell here, and the chromosomes are made up of chromatin networks. Chromatin means DNA along with certain proteins called histone proteins. There are certain proteins called histone and non-histone proteins which try to wrap around the DNA molecules, and when they wrap around the DNA molecules, they get fit into the nucleus. This is how the chromosomes gets condensed. When I say condensed, they becomes tight. How do they become tight? The DNA gets wrapped around the histone proteins and it is placed into a proper structure called chromosome. There are around 46 chromosomes in human nucleus, and each chromosomes has certain genes, several genes actually, and each gene can code for a particular character. Today we all know that the chromosomes carry genes, isn't it? The chromosomes carry genes. But in those days, when Mendel's work was happening, that is in the 1900, when the three scientists rediscovered the Mendel's work, nobody was knowing that the genes were present or located on the chromosome. So this theory, that is the chromosomal theory of inheritance, this theory talks about the location of genes on the chromosome. That is how exactly the genes and the chromosomes are linked. We'll be understanding in this particular session. Moving on to this, it was Mr. Walter Sutton and Theodore Boveri in 1902 who proposed this particular chromosomal theory of inheritance. We have Walter Sutton here and Theodore Boveri here. These two scientists independently they were working, and they together proposed it in 1902, that is the chromosomal theory of inheritance. What did they see, how did they propose it is, when they were trying to work out with the chromosomes, they related their studies with the Mendel's work, that is the factors or the genes what three scientists, that is Hugo de Veris, Carl Correns and Teschmark, what they had published. These two scientists were trying to correlate with this, that is the chromosomes movement and the Mendelian factors movement. So what is the similarities or what were their observations is, here are the two main observations that they have seen here, that is separation of homologous pair of chromosomes, and the segregation of Mendelian contrasting factors both happen during the meiosis. This is what was their first observation, that is the homologous pair of chromosomes also separate in the meiosis, and also the segregation of Mendelian factors also happen in the meiosis stage. Both these particular inheritance characteristics what we have, that is the Mendelian factors and also the characters coding chromosomes, both do move in a same phenomenon in the meiosis stage. Next, the second observation what they had made is, that is independent assortment of homologous pair of chromosomes were also happening, and independent assortment of Mendelian contrasting factor also was happening in meiosis. In the first observation, they have seen the separation of both chromosome and the Mendelian factor. In the second observation, they are talking about the assortment. Assortment is movement. First, they separate together. That is the chromosome and the gene both separate in the meiosis. In the second one, the separated chromosome and the genes, they assort, they get moved to different gametes again in meiosis. The movement of chromosome and the movement of genes are quite happening at the same time. So that is why they say the chromosomal behavior is equal to the gene behavior. Both gene and chromosome behave normally the same. There is another example I could give for you. That is, if you take an example of the sperm and the ovum, we all know the structure of the sperm and ovum are different. Sperm is very minute, ovum is quite large, and sperm shape is also different, the ovum shape is also different. But students, I'll make you understand here, that is the sperm also carry 23 chromosomes and also the ovum carries 23 chromosomes. Both are haploid in nature, that is N and N, each one carry 23 and 23 chromosomes. When they both fuse, we get a zygote of 2N. In this 2N, we get around 46 chromosomes in the zygote, isn't it? So this gametes, when they are formed, imagine we have capital T here, we have small t here. This is the same thing what we have studied in the monohybrid cross. These two alleles move separately to two different gametes, isn't it? Like this. That is, only one allele will come to this one, the other allele will come to this gamete, isn't it? The same way, the 23 chromosomes get separated to two sperms from the father, and the same way the 23 chromosomes will be separated to 2 ovumes of the mother, and one will be denaturing there. That is another story. But then here each gamete will be having only one set of chromosomes, isn't it? So here you could see that gamete has only one chromosome as well as one Mendelian factor. That is, any gamete you take, if you take the chromosome number 1, the first chromosome is here in the one gamete. In the next gamete only the other chromosome 1 will be there. So this chromosome which is moved here will be having an allele, that is nothing but the Mendelian factor. Out of the two homologous pairs, the allele will also come out of these two alleles. If you could see here, out of these two alleles only one will come to one gamete. Same way, out of 46 chromosomes only one set moves to this gamete, isn't it? So there is similarity between the chromosomal and the gene behavior. Next, after this observation, what Sutton and Boveri has done, we'll go into the theory what they have published. So here is the thing, that is, according to this theory, genes are hereditary units. Genes are hereditary units which are located on the chromosomes. See here students, you have a chromosome picture here. In this chromosome, this is a particular segment where exactly the genes are present. These genes which are present here are the hereditary units which are located on the chromosomes, and chromosomes carry the genes from one generation to other generation. We say genes move from one generation to other generation, isn't it? So the one who is carrying the gene here is the chromosome. So these chromosomes carry the genes from one generation to the other generations. I hope you understood the first postulate here, that is, genes are the hereditary units which are located on the chromosomes, and chromosomes carry the genes from one generation to the other. The next one, the next postulate of this particular theory is chromosomes undergo separation from homologous pair and then assort independently along with genes carried by them during meiosis. Students, this postulate is the mixture of both law of segregation and law of independent assortment, if you could see properly. There we spoke about the alleles separate and the alleles get assorted independently in the Mendelian genetics, but here we say chromosomes undergo separation and they assort independently from the homologous pair, right? And while the chromosomes are moving, they carry genes together in meiosis. This theory is the one which is linking both of genes and with the chromosomes. Here we have another picture showing that the chromosomes are those which are carrying the alleles. We will discuss this picture broadly here. That is, we have taken an example of the same dihybrid cross. If you remember the dihybrid cross, we have taken capital Y and capital R for yellow round seeds, and we had taken small y and small r for wrinkled green seeds, isn't it? The same thing what we have discussed in respect with alleles in dihybrid cross will be discussing here in the movement of chromosomes. Consider we have a chromosome here with capital Y, small y and capital R, small r. This particular chromosome is carrying these alleles here. The white mark, if you could see here, this white marks are the loci where exactly the alleles are present. Now what happens is, when this particular cell which is carrying these alleles undergoes replication, that is, the chromosomes undergo replication, that is nothing but the doubling or the duplication of the set of chromosomes here. Each one becomes double. The capital Y here will become 2, the capital Y and the capital Y. The small y also becomes 2 here. Same way the capital R becomes 2 and the small r becomes 2. This way the replication has happened here. But we'll take another case. This is the possibility 1. This is the possibility 2. In the first possibility, what we have taken, capital letter Y with capital letter R and small letter y with small letter r. But here we'll take the capital letter Y. What if it is moving with small letter r? The small letter y, what if it is moving with capital letter R? We are taking two possibilities to understand how exactly the gametes are formed and wherein the genes are carried on the chromosomes here. Now in the first possibility, we'll see here Again here with the meiosis 1 happens. Students, you have to remember the metaphase and anaphase stage. In the metaphase, first the chromosomes are getting aligned on the metaphase plate here in the equatorial region, but when they get segregated, when they separate exactly, the segregation actually happens in the anaphase stage. That is, the chromosomes move here like this from the centromeres, isn't it? And the chromosomes also move here into the another opposite pole here, right, isn't it? So that particular stage is here, meiosis 1. In this particular meiosis, the half of the chromosomes get separated. That is, capital Y comes with capital R and small y comes with small r here. But in the second possibility, what had happened, the capital Y has come with the small r here, and here the small y is coming with the capital R. So all the 4 possibilities we are checking out here. In all the 4 possibilities, what happens is, after the meiosis 2, after the next stage, that is in the meiosis 2, if you see, the capital Y and the capital R goes for one gamete, and another capital Y and another capital R goes for another gamete. Similarly, small y, small r to one gamete, small y, small r to another gamete. Here capital Y and small r comes to this, and capital Y and small r comes to this. Similarly, we have small y and capital R, small y and capital R. In this, what we have understood is, we have got 1/4 of the gametes containing capital Y, capital R. We have 1/4 of the gametes containing small y, small r. We have another 1/4 containing capital Y and small r. Another 1/4 having small y and capital R. In these 4 possibilities, if you remember the Punnett square that we have drawn for the dihybrid cross, we had written the 4 different gametes here, isn't it? The 4 different gametes that we had written are the same. Capital Y, capital R, small y, small r, capital Y and small r, small y and capital R. The same gametes we had written. But here what we have done, we have located these alleles on the chromosomes. That is, all these alleles or the genes are located on the chromosomes, and the chromosomes are those which will be separating, and they are assorting independently during this meiosis stage, right? This is what is chromosomal theory of inheritance. We have to just relate with gene and with chromosome. We have to say the chromosome movement is not different and the gene movement is not different. If the chromosome is moving, the chromosomes are carrying the genes and then they are moving. So both the movement of genes and the chromosomes are correlated. This theory suggests this, and it was given by Boveri and Sutton. Next, we have another person here. He is Mr. T.H. Morgan. He is Thomas Hunt Morgan. What happened is, when Boveri and Sutton, when they gave the chromosomal theory of inheritance, this scientist, that is T.H. Morgan, along with his group, that is his own student, that is Student, Muller and Bridges, all these 4 scientists are those who have contributed a lot for the field of genetics, but after Mendel they have worked. This chromosomal theory of inheritance that was published by Theodore Boveri and Walter Sutton was being confirmed by this scientist T.H. Morgan. This fellow T.H. Morgan was very much fascinated with Drosophila melanogaster, the fruit fly. The fruit fly, Drosophila melanogaster, he has worked with. Just like how Mendel was very much fascinated with the garden pea plant, T.H. Morgan was very much fascinated with the Drosophila melanogaster, the fruit fly, and he has done extensive work with Drosophila. He has studied something called linkage, sex linkage, where Bridges, Muller, all these scientists come to the picture there. All of them have given a lot of contributions to the field of genetics. We can make another session for that, but here now T.H. Morgan has proved this chromosomal theory of inheritance, that it happens, and whatever Walter Sutton and Theodore Boveri has told is 100% true. He has worked with Drosophila melanogaster, the fruit fly. Why? Because Drosophila melanogaster is a model organism. We consider this as a model organism because we can easily differentiate between the male and the female opposite sexes. Here the small Drosophila, what we have given here, it is a male Drosophila, and the lengthier one is here a female Drosophila. Here males are usually short and females are usually lengthier, and males have the curved darkish abdomen, whereas the females have the pointed abdomen here. Also Here these Drosophila flies, when they fertilize, when they fuse, they give a lot of offsprings. Their lifespan is usually very less, that is around for 2 weeks their lifespan is, and the media where the Drosophilas grow is quite easily prepared also. It is very easy to handle them in the laboratory. So this person, T.H. Morgan, was very much fascinated with this flies, and he also found certain mutant copies. Certain mutant copies are nothing but, I'll take an example. One of the Drosophila what Mendel was working was always having the red dye, few of them, but so when he was working a lot of with Drosophila flies, he also found a whitish colored Drosophila fly also, and he has worked with that particular mutant types and all, and he is the person who has confirmed this chromosomal theory of inheritance. Next, the same T.H. Morgan has given the theory for linkage also. We'll see that particular concept in the next session. Thank you everyone.